Today we will be talking about ampliCan. This is an R package, of course, for automated analysis of CRISPR experiments. And this package is designed to analyze amplicon sequencing data. So amplicon, you can see the name ampliCan because amplicon sequencing. So anything that has, but not only practically, this package can also analyze other types of data than amplicon sequencing. But the underlying theme is that it requires you to have like primers. It assumes there are PCR primers. It can be fake primers if you have, for example, nanopore data or some other type of data. And then you can pretend that you have those primers. And then we go to the amplicon sequencing. So like if you Google this amplicon sequencing, I don't have really a presentation ready for today. I didn't have the time to prepare it, unfortunately. So it will be a little bit back and forth. Hope you can see this. This is just standard PCR that we use to grab. You need a forward primer and a reverse primers on both sides of your sequence. Like I have prepared this kind of tool to help you visualize how amplicon works. So we need to have a forward primer on a piece of a genome and then a reverse primer, and then the sequence in between them will be multiplied. And this is what we sequence, right? And normally reads have it from the forward, and this goes the read goes from this side to this, and then we have it also from the back, reverse sequenced from this part to this part. And yes, I prepared a couple of extra resources which you can use to because I figured this is an opportunity to sort of go back to ampliCan, which was published many years ago. I think it was published around 8 years ago. This is the ampliCan website on Bioconductor. There should be somewhere visible how many years it's available. So I recommend normally. Yes, I prepared also some outline of what we will be talking about. So first, we will go through what is ampliCan and why use it. Then we will go to how to install it from GitHub or from Devel or from Bioconductor website, how it works. We will go through some example data. We'll talk about how to create the most important file for ampliCan, which is the config, how to run it, and then how to tweak the results, how to interpret variant plots, and then how to adjust the resulting reports. And then some maybe special hidden functions in ampliCan, and then some questions. Or you can ask me questions during the presentation. So let's start with what is ampliCan, right? So this is an R package, we know that. But why should we use it? So I have rerun one of my benchmarks. This is the original benchmark that I just recently have rerun. And you can see here, maybe I can make it bigger. What you can see here is that I'm simulating, let's move this also. I'm simulating different CRISPR created deletions or insertions and mismatches of different sizes. And then I compare the output of different tools that can analyze the CRISPR outputs, assuming this is from amplicon sequencing. So you've got the yellow, it's ampliCan. We've got the CRISPResso, which is very popular Python. We have the CRISPector tool, and we've got this NF-core CRISP-seq. And I have just recently rerun this, like yesterday. What it does is we know the ground truth because we know that we have I injected also. This is simulated data, so I inject deletions. So I know how many I injected. So I can have so then staying on the line is perfect. So when we have zero deletions, zero CRISPR efficiency, then we are on 0, and all tools are correct. But we only need like 33% of different kinds of events to start see the difference. And I honestly have no idea why CRISPR performs so poorly on my benchmarks here. I will try to figure it out. I assume I know only why CRISPR-seq doesn't do well, this NF core. So NF core has the issue that it rejects reads that have insertions. So all the kinds of indels, insertions, they're gonna be rejected, and then it causes those some samples, some gene samples to be completely wrong, right? But this is only for those insertions, especially if they're quite big, more than 10 base pairs. I can see that ampliCan has also some issue, and I have investigated. This is gonna be fixed also. This is due to the filter on of targets, because there is also an off-target filter, and it kind of mistakenly detects some of those as off targets. And so most of the tools are struggling with insertions. But luckily for us, CRISPR usually doesn't introduce insertions. It mostly introduces deletions, right? So, but even in those cases, CRISPR doesn't really do well on my benchmark. I'm not sure why it is like that. But this is kind of worrying to me because the tool is very widely used. So at least in ampliCan, I know what I get, sort of. So I recommend using that tool. So this is the benchmark that I will be working on. This is sort of preliminary results, let's say. So now let's take a look at how ampliCan actually, how to install ampliCan. So you can go to Bioconductor website. This is the development version here. And you can just simply copy this and run it in your command line in R, and that will install you all the dependencies you need. But I recommend to also install yourself a version from GitHub. After you do that, you can install yourself a version from GitHub. So this is GitHub here. You can also use a link over here. There is normally this is a link. You see URL. You can go here. And you can also, if you have issues or some bugs to report, you can comment on this under this link. So at the moment, nothing is open, everything is closed. And here you can see the latest version. I just updated 3 hours ago this package. So it will take maybe 2 days or around 2 days for Bioconductor to sort of rebuild it on here on Devel. On the branch, and then it will become the actual branch in maybe in like a couple months. So it is a little bit lagging behind for the development. But yes, so you can always install yourself from here and then install from GitHub by doing git remotes install. You can open RStudio. And you can always do like remotes install git, and here type just Valen lab, and then you're gonna have the latest version, and then you can check which version you have here in the packages. So I have version 1.35.4. So ampliCan doesn't really depend on any very changing packages, so it's okay to install this from the GitHub. So because most of the code is sort of self-contained. I try to have very little dependencies. You can see what we depend on in the description. You see, it depends on the basic. This usually never change for Bioconductor. These are like very basic Bioconductor packages that never change. And then some extras that also are sort of core of the Bioconductor which rarely changes so that it's not backwards compatible, so it's safe to install from GitHub. Okay, and so now we know how to install this package, and then we can try to talk about how really it works. So you have also a couple of resources on this ampliCan website. And so if you go to the development version, so if you are in the normal Bioconductor, you can click here the version for the devel. And in the devel version, which I have open over here, you have vignettes. So first we've got the standard frequently asked questions. We've got the overview, this is sort of the main vignette. We've got the frequently asked questions. We've got the latest ones that I have added. They are not yet available on the normal normal Bioconductor, the main one. This is only on devel. So I have added one resource, which is code explained. So this is about, we'll see in a second, and then how to prepare config with the use of large language models, because this is very convenient way of preparing the config. And then we have one how to tweak the output of ampliCan. So this is, and then the one here, these are example reports. So these are the default reports that if you run ampliCan on examples, this is what you're gonna see. So that they are sort of copied over as the vignettes. So we can open, this report. This is sort of, you can take a look what ampliCan is giving us at the end of the day. So this is like a summary. We're gonna have all kinds of plots and links to other reports. So we can click that and see by ID, for example, this report. So this is like automated reporting. I think ampliCan is maybe the only one that has it so extensively done, so that you have, you know, that they have also increased less some automatic reports, but they look entirely different. Okay, but going back, something fails. Some worked. So we have the one where code is explained. I just created that one recently. So this one is useful if you don't know how to read R code too well. It's like a reference of everything, every single step of the pipeline. So what is happening on each step of the pipeline? So we've got the setup, we've got the reading and quality filtering, then it's collapsing identical reads step, assigning reads to the experiments, aligning to the amplicons, there is HDR detection step. This is about the class for the alignment experiment set, which contains sort of ampliCan experiment, and then how to extract the events, filtering, how to do consensus, cut site detection, then shifting coordinates, and then normalizing against controls. This is very important. Summarizing, and then how reports are created, and then there is even how to use external alignments. So you can even do alignments with other tools than ampliCan, and then sort of process the rest with ampliCan. So it's possible. But this was sort of hidden functions until I did this here, and then it's about plotting and visualization. So this is a useful reference, zero code, which is sort of, if you want to read a little bit how it works, this is a very good reference. You don't need to read the code line by line, but just read this if you want to. It's not that long, I think. So then we have also how to prepare a config with the, if you have it from in form of Excel or some sort of table, and use a large language model to assist you. Because I noticed that the biggest struggle to use ampliCan is when people have to prepare the config file, right? Config file defines all parameters. It defines which sample contains which guides. It also contains which sample is on control, which sample has which primers, how to demultiplex each sample. So config is kind of like a single table where everything is defined. And I guess this is a bit different than other tools because ampliCan is just a table. But it's different table than usually what you get from biologists when you need to analyze this data. So this file is prepared to use large language models so that you don't have to do this all by yourself. And it's much easier to do. I think these models are useful for transforming Excel table into format that is what ampliCan expects. And then we have also about tweaking the ampliCan output. So you can, ampliCan has tons of options and you can adjust a lot of different knobs. And that means this is also not always easy to get the result you want, or rather you can do it in multiple different ways with different thresholds and then get better results. And this is a file that I made to help you tweak the output. Normally, when I work when I analyze some data, I'm trying to tweak the output multiple times in different ways, depending on what I see on the plots. So we'll also discuss that. Yes, and then you can also play around with just editing the reports themselves. So this is also a useful trick for whenever you want to adjust some plots or you don't like how it looks, you can change everything there because this is just normal, regular RMD, which you can need whenever you like. Okay, let's see whether this finally works. And this one worked. So this one is ampliCan overview vignette. And this is the first one you should probably look through. And it shows you what is the big picture of ampliCan. This is the big picture. Oh, again, this is probably, yeah. Okay, so this is how the functions are sort of structured because ampliCan goes through this flow. Everything that you can run, like as a main function called ampliCan pipeline, which you can run, it takes simply the folders, paths, and it takes a lot of parameters, and it takes the config file, and then it produces everything for you, all the reports and everything. So, but it goes through all of those steps, and you can yourself tweak any of those steps at any point, right? So you can also build on top of ampliCan and build something more useful for you, something custom. So ampliCan pipeline will take FASTQ files where they are sitting in the folder, and it will take the experiment description in form of config. And then what it does, it checks the description file, and then it splits everything into barcodes. So I know majority of you probably don't have the issue of having in one simple sample multiple barcodes. But many people do. And when I created ampliCan, that was for this use case for people in I think it was Broad or some other university in States where when they were basically pulling many different samples together, putting them all into with PCR, and they were basically having different forward and reverse primers. So ampliCan is splitting based on forward and reverse primers into different IDs. And then we've got align. So this is the step where ampliCan is performing the alignment. It's doing this completely differently than any other software because it's doing Needman-Wunsch, and this Needman-Wunsch is based on a PW align package from Bioconductor. It might sound a little bit weird. Why not use existing aligners? But I think the reason for that is that I get a good control over the alignment settings. And the alignment we have here is kind of specific. So CRISPR alignment is different a little bit than normal alignment. We want to have the best possible alignment, I think. And here we can tweak every single detail. So if you want to, you can look exactly which parameters I used, and you can also supply your own parameters for alignment. So in here, if I load ampliCan, it also recommends you to cite the tool if you have the opportunity. And then if you do ampliCan align, you can see the question mark. Okay, and this is. Then you can see here that you can supply your own scoring matrix. And you can supply what kind of gap opening and gap extension you want to allow, and so on. So, and the same goes for pipeline, for the main one. But yeah, so you can see here you can supply scoring matrix and gap opening and gap extension. So I, on purpose, I punish opening a gap. We don't want, because this is CRISPR, we expect a gap. And normally, aligners don't really expect that. We expect a gap, and we want to actually make sure that the gap will be, there will be a single gap, preferably, instead of multiple gaps. So we want to punish gap opening so that the alignment actually knows the gap is there. And then we've got the standard. I think this is standard, but just slightly adjusted. So this is like an array of different bases. So I have this kind of scores by default. So there's many different options in ampliCan, which you can adjust. But of course, defaults are supposed to be sort of working start, at least for the normal cases. Okay, so this is about the alignment. Then it is quality filtering, grouping similar reads together. So this is all what happens inside the ampliCan alignment. So there is quality filtering first before the alignment. There is a grouping of similar reads into the same sort of bucket. We are matching primers to the reads. Yeah, and it says we are using Needman-Wunsch with optimized parameters. And this creates this alignment experiment set. So this is like an object which you can reuse. And it has a couple functions. You can look up the alignments. So it's possible to print the alignments and look them up, whether you are happy with them or not. You can also write the alignments to just different types of files to see whether they are correct or not. And you can merge multiple different alignment experiment sets together if you want to. But normally, the main pathway is to extract events. So what I mean by events? Events is insertions, deletions, and mismatches inside the reads. So this is another weird quirk about ampliCan, which I will talk about later. ampliCan is working on event level instead of read level. And then you do the overlap checks because we want to know, does it overlap the guide or like region of interest or not? Then we want to know the consensus because ampliCan is aligning forward reads and reverse reads separately. So then it has to sort of build a consensus from forward and reverse reads. And then we have filters for primer dimers and other kind of noise. And then we've got just shifting and normalization. So normalization against the controls. That leads us to summarization, where we can actually just calculate rates of all kinds and then build those reports. And some plots are using those events, right? So that's why there is arrow leading to those plotting functions. And this is supposed to be compatible, and those formats should be compatible with genomic ranges, with data table, depth. So there is no fancy formats here. It's really tables. I try to make it all tables. Okay, so this is the introduction to ampliCan. This is our consensus. We'll talk about it later. And this is config file, the example one, which you can see. So in the examples you can just run the system file xdata. So if I go here, if I run this is where my example config is located. So I can read it this like that. First, and then we can go like this. And then in our environment is this table of the example config. And you can see here exactly what is expected of you in the config. We'll go through this in a second. Okay. It's a little bit chaotic, but it also, you have this description of what is needed in the config here. And then we will go through how to build the config using those large language models also. Right, and then you have the default options run. So all you need is really this. You specify where is my config, where is my fastq folder, where do I want the results, and then you just run it, right? ampliCan Python. And it will take a bit longer than other tools, but I think the results might be worth it. And there is, a small overview of the files that are created during the analysis. You can see all the statistics like filters, bad base quality, bad average quality, bad alphabet, unassigned reads, very important. And then there is summary. So summary is usually the same as the config file, extended with additional columns, which are like how many reads were there, how many reads were filtered, how many reads had insertion, deletion, how many reads were quantified as edited reads, and how many reads were frame shifted. And then from here, you can build your fractions if you want to plot them. And then there is also run parameters txt file, which contains all the most important parameters of the package that you specified during the run, so that you don't get confused. This is my result from somewhere, and what was actually being run as options. You also get alignment folder, and the alignment folder contains, of course, all the alignments, and you can also load those. This is the table of the events, right? So you can see, okay, this read had a replacement, so this is an insertion, and the alignment score of this read was this high, and this read happened 3 times, and this is not homology directed report, so it's not quantified as such. And then does it overlaps means there is another like that. This overlaps our region of interest, so the guide location, and then consensus that this is confirmed by the reverse reads. And then you can also see the alignments. If you look up alignments, you have an example how to do it. And so you can run this and see this type of output of the alignment, you see? So this you can see, okay, primer would be somewhere over here and over here. And then the read goes, we can see sequencing length is 115. And then you can see the alignment goes like this. So this is very large insertion. And this is kind of clean alignment. So first we've got deletion, and then we have the insertion. And then you get the reports also, of course. And each of the, you can disable making of the reports just as an option in ampliCan, and then it doesn't make them immediately. You make them yourself if you want to, because it takes very long time to actually, most of the run time is this, meeting the reports. And then some details about which functions you can use, and some details about normalization. So this is the overview, but we will go into much more detail in all of this. And then we've got also the vignette. This one didn't load. Yes, now it did load. So frequently asked questions. So can the ampliCan be used for TAR in case or other thing? And the answer is, of course, it can. It just depends on the primers. So even if you don't have the primers, you can always sort of input the sequences as primers, so like fake ones, just something that, because the idea is to anchor. You can actually do a lot of controls. We'll go through that. And these are some other questions that people are asking. What can I do when I don't have complicated files, right? So if you don't have complicated files, you just have a smaller config file base. So many of those, the software works. You will see in a moment. Okay, so now you know where to find all available already online on the devel branch. Or you can just grab it from GitHub if you need it. Okay, so now let's go. I made this sort of let's say presentation on how it works really. So the idea of ampliCan is that you will have forward primer and reverse primer. This is what you'd use for PCR. It's good if you sequence. This is assumed that your guide is somewhere in the middle, preferably perfectly in the middle, because you want to have, when it cuts, it will make like a deletion, if you imagine. Like if this region is deleted, and then you see this in sequencing read, it will go from this side here, and you need to capture preferably both forward primer and reverse primer in that deletion. But sometimes deletion happens so that it is very large, and it goes from here over there, and it deletes also the primer. And then you are no longer so sure this is correct, right? So you have only, this only this part. So this kind of reads we are rejecting in ampliCan. So this is what you want. You want to have guide sides, and you specify that normally in the config as the uppercase, and then or the cut side that you expect. So if you have, for example, Nikkei, so you have two guides, maybe here and over here, then you can specify everything between them as the uppercase, and then this region will be considered as the overlapping region of interest, right? So everything you put in uppercase in the config file. Where was the config file? Example. Here is the example of the config file. So you see here, in uppercase letters, you have the guides. I normally put the guides in uppercase. And then I add the cut buffer. So it adds additional 10 bases on each side of the guide for my region of interest. And this is how I specify ampliCan to work. For donor, we don't have to put that. It doesn't matter at all. Donor is if you are doing homology-directed repair, you can specify the donor. Otherwise, you leave the donor column empty. And for donor, this is the template sequence that you are expecting to find. One important detail is that donor should have the same primers free. It's best if your amplicon sequences are starting with a primer. This is a primer, and then they end with a primer. And then the best is if this one can also start with a primer and end, so that the only difference is what you expect to input there, or like find the extras. Maybe you are fixing single base or something. Okay, I think we can move on to our presentation. Sorry, not this one here. Right. So the idea is that we find those primers inside your reads, and this is what we are using for matching barcodes. So going back, maybe I will do it like this, and we go back here. So here you can see what I specify in the config. IDs column is just a column for something unique named for identifying this particular line inside the ampliCan config. Barcode means grouping, usually between the FASTQ files. So here's an example of multiplexing, where you have single FASTQ file. By the way, you don't input full path to the file, just the name of the file. The path you input as the when you run the ampliCan pipeline function. So this one is the same as this one, right? So this file contains two different primers, this primer and this primer, paired with this primer and this primer. And this is probably the most confusing part. So this is gonna do the multiplexing step. And then we have two different barcodes, so to say, which have unique inside each barcode. You need to have unique combination of forward and reverse primers, because this is what is being used to recognize and split reads into samples. So I will here, my read comes in, and I match using ampliCan towards this read. Does it contain forward primer? Does it contain reverse primer? And then it checks the option. There is an option for that in ampliCan. Let's see, it's over here. The option is called FASTQ files. Then depending on the FASTQ files, if you have option 0.5, that means finding either forward primer or reverse primer will quantify the read to this ID, or this is or option. This is the default. This primer or this primer, because normally people have them unique, like we have in our config over here. See, this one is unique, this one is unique, and this one is unique. So just grabbing one of them is enough to know this belongs to this ID. Very often, reverse primers are a little bit botched, or maybe reverse reads are bad quality, and then it is difficult to quantify those primers. I have an option in ampliCan where you can specify how many mismatches are allowed inside the primer. So the default is 2. So you can adjust this if you have more like different setup for the experiment. So if you specify, here is FASTQ description, FASTQ files, you see, 0, that means we are using both FASTQ files, and at the same time, both reverse and forward primers have to be there. If you use 0.5, then just one of them has to be there. So you can use this when you, oh yeah, this is also useful if it was trimmed for you. Maybe you have reads where primers were trimmed, for example. But you need at least something to deconvolute reads, right? One FASTQ files, one means you only use FASTQ files for the forward and two for the reverse. Yeah, and that's it. And then this combined with primer mismatch number allows you to kind of manipulate the multiplexing step. And the output of the multiplexing step will be always visible in those ports called barcode, the port by barcode. So when you run report by barcode, very slow internet because it's not nice. They were split basically, and also you can see where at the highest, maybe when it loads in a second. I wonder what is the problem here. We can maybe take a look at some of my files, like here. We've got a real experiment of someone that I analyzed, and here we have the barcode report. Here you can see what is inside the barcode report for real experiment. So in this case, you can see which groups. Oh, this is very simple experiment. There is no real demultiplexing here. It's just this barcode has this control. This one has this fix, and then with some knockouts. And then you can see the how reads are distributed across different barcodes. How many reads are filtered, the edit rates, and so on. And then you can see the most useful, I think, is top unassigned reads. If you have difficulty demultiplexing your reads, it will be here. You will see it because the frequency will be high for that for those reads. And then you've got P1 and P2. So P1 is forward read and S1 is reverse read. And you can see how it aligns together, forward and reverse read. This is to check whether you have something messed up or whether these are real reads. And this gives you an insight into how well reverse reads are overlapping with the forward reads. So here you can see that our 150 base pair sequencing does not fully allow to capture the full extent. So if we go back to our here view, so for very often this happens when people design their sequencing experiments, they're gonna put 150 base pair from here to here is gonna be 150 base pair. And from here to here is gonna be another 150 base pairs. So they want to capture 300 base pairs of information with 150 base pair sequencing. That's a mistake because normally that could work but for CRISPR, if the cut is here, imagine the cut is being done here. That's gonna give us confusing view from the perspective of my forward and reverse reads, because this read will just terminate over here. You don't know what is happening. This read will terminate over here, and you have no confirmation of the end, like what is after. And that is the most useful. If you can somehow capture this part of the read and then this part of the read, that is the most useful, because then you know you have like two parts of two sides of that deletion. Okay, I hope this is understandable. So yes, in this case, this is useful for that. This shows you because these reads are not assigned anywhere. I don't know which amplicon to align them to. So I align just read forward and reverse, and you can see that there is many reads like that. In this case, it is almost nothing. So in this case, almost everything was nicely multiplexed, right? Because we had very clear cases. All reads belong to this single sample and so on. How it looks like for our example, did it load? No. I think this is some bioconductor issues, maybe. Okay, going back to our this presentation. So I think we can understand now what happens with multiplexing. So when you specify the reads, sorry, in the config file, when you specify in the config file, where was it? Here, you specify those forward primers and reverse primers. And this is the most important part for the multiplexing. So for every single pair that you have in the file, you need to have another row, and that's it. So you input as many rows as you have to for these combinations. Okay. Furthermore, you need to specify a guide. You don't have to actually specify a guide, but then you're gonna get a warning, and it doesn't do anything. It will still proceed to analyze if it doesn't find your guide inside the amplicon. But I think it is a useful check that the guides inside the amplicon will be found, and then the direction says in which direction is the guide. So if this is the opposite, so you can guide is GGGGG for G's, the direction is 1, and here uppercase is CCCC. So that means the guide is the opposite direction. Okay, the amplicon. This is just a simple check. Guide RNA column, it has two purposes. It is a check for if do we find it in the amplicon, and the second purpose is that we produce this guide related metaplots. So we will see that later. And then another thing is that basically another confusion in this is the control column. So if you don't want to do normalization, you just specify everywhere false, and it is fine. It will just not normalize anything to anything. Okay, let's move on to the next step. So this is the first step. Then we have the quality scanning. Of course, that's normal. So checks every read whether it passes the quality or not. And you can tweak those options for quality. So there are inside R, the quality options are here, minimum quality. So the default is 20. So massively, most of your reads will go to this. And then average quality. Normally, we don't filter on that by default, but you can set it if you want to. So these two are defining the quality filters. Then we've got the next step is collapsing identical reads. So this is only because we are doing the alignment with Nid-Banouche, and it takes very long time. So we prefer to collapse identical reads and then do the alignment and then sort of deal with the fact that we need to remember every single time that the count of this read is higher, right? And also I assumed that when I was creating this, I assumed that because CRISPR is cutting, it very often makes very similar cuts, but also just the regular controls will not have very unique, high uniqueness in read counts. Normally, they will have almost all reads the same, right? If you have control. So then that saves a lot of time when doing the alignment, because then you are like aligning one read instead of thousands, which are identical. So this is the read collapsing. And then read collapsing, this is what is then visible in the column count. as counts. Okay, so now this is this is finding inside the reads and assigning. So we had animation for that. You can see here, forward primer was found, reverse primer was found. So these reads are assigned, the same for this one. When you don't find it, these reads land in unassigned reads. Right, we talked about it. And then when reads are unassigned, then we have the issue. Usually we have to check what is going on. From my experience, maybe around 10% of unassigned reads is already quite high, but if you can get it to like 5% or something, then that's really good. Okay, then we have this information that primers are the ones that anchor the alignment. This is absolutely the most important and crucial thing for AmpliCan. So you can see here, if you have no edits, you should expect something like this, very high score. And if you have some deletion, then you can expect our score to be lower. And also the deletion should be here, right? In the middle, somewhere where the guide made it. And yeah, we preferably want to have a chunk of the primers on this side and on this side. Okay, then we have the HDR quantification. So this is the part where you have also kind of complicated setup in AmpliCan, because this was added after I published the package, and then I continuously iterated on it. So what we do is we are performing two alignments. One alignment is towards the amplicon sequence. We grab the read, we align it towards the amplicon like normally, and then we do the same, we align it towards the donor. And then we check, and depending which kind of options is specified, and we try to mark those reads as homology directed for per read. And so we've got special options for that. And the options are here, donor mismatch, and then donor strict. So some cases, you want only the perfect HDR. So in that situation, I specify the strict. So it only allows the reads that contain every event that you wanted to have inside your. So basically, these have to.
We lost you, Kornel.
Oh, my connection is a bit unstable, it seems. Can you hear me?
Yes, we can hear you now.
Yeah, okay, good. Okay, so getting back to this, we've got the donor strict, which is the option which you can, it just basically makes the, you need to find all kind of mismatches, whatever was found in the because we also align also donor sequence towards the amplicon, and then we can find all kinds of events like, okay, this is a mismatch maybe. And the all difference between donor and amplicon is this mismatch event. And then we are looking for these events, and they all have to be present if you select that as donor strict. Then this option, donor mismatch, is to allow some sort of mismatches around here, because like, sequencing isn't perfect, calling the bases isn't perfect. So usually we have some sort of like a mismatch somewhere, and then we want to count this or not. It's up to you. You can specify this with donor mismatch. This only works if this donor strict is false, because donor strict means everything has to be perfect. Okay, like you are maybe editing human cells or like patient cell lines, and then you want to just quantify how many perfect edits are there. So then you can use this donor strict. Okay, so this is the HDR, and then we mark those reads that are detected as HDR, basically, and then they are being summed up at the end. and they are in the table. So this is this step. Then this is the event extraction step. So this is another differentiating thing of ampliCan and other tools. Because in ampliCan, you have, like we are operating on the event level, not read level. So imagine this situation. So here's an example. So to me, first of all, what is an event? Event is a mismatch, deletion, or insertion. And I call this events because I assume these are events caused by either a background, like genomic variants of a patient, or cell line, or maybe this is a CRISPR-made event. And then the idea is to sort of grab read events and start comparing them on the event level, not on the read level. So on read level, it means it's like you are comparing maybe this. You are not sure what is this, right? Is it a CRISPR edited sample or not? So then that changes the quantification step. But in here, I expect event to be here on this line because this is where CRISPR sits. So that makes ampliCan a little bit more tweakable. And you can play around with those events. Like you can do HDR, like I do. Okay, we wanted to have HDR with this small insertion here and this deletion. So that means we are checking for those events. We only quantify reads that are having this and this. And maybe we will allow a mismatch over here and mismatch over here. It doesn't have to be perfect. So this is another very important thing in ampliCan. So we are operating on everything on the event level, and we try to kind of work with events. And this is useful for all kinds of quantifications, because this is sort of this is like doing variant calling without really doing variant calling. It's like you could call it some sort of some way of variant calling. So we call those variants in our reads, and then we perform operations on them. And we store variants in a table, basically, to make things simple. But these are not as complex as normal variant caller with all possible structural variants and so on. And then, yeah, the step for removal of artifacts. So first, if an event overlaps primers, it is removed. So if you have a deletion somewhere over here, that is probably some sort of an issue for us, and we are removing this. Then you've got primer-dimer cases, which means basically this primer connected to this one. This is difficult to quantify whether this is a CRISPR-made event or this is just a primer-dimer. And then another filter is this low-quality reads, which uses this Clara clustering. And it's, well, you can disable that. By default, it is on. But you can disable this easily. There is an option for it. Over here, you've got an option called max remove filter LQR. So you can, for example, set this to 0, and this is basically disabled. Oh, sorry, you have also event filters. You can set event filter to false, and this is then, yeah, disabled. By default, it is on. And how it works is that we have the scores of aligned reads, and we can put it on a plot. Here scores, and here the number of events. And it basically clusters the reads, and it tries to detect how many clusters of reads we have. If you have normal, we expect 2 clusters. For normal samples, treated samples, we expect a cluster of unedited reads and a cluster of edited reads. But if there is some sort of messy weird reads, that they are the treat cluster. And this is what is removed by default here. Okay. Then there is the step of quantifying the reads. So exactly for that, we are using the uppercase region.
You're breaking again. Sorry, Kornel, we lost you
yeah,
there.
I have internet, it seems. On the previous slide, here.
Yeah, you can start
So
there.
yes, let me repeat. Yes, so if you need to specify your region of interest with uppercase in Amplicon, and that is what we can quantify as the interesting event. Right, and then it's shifting. So we are shifting towards the most left side. This is only useful for making metaplots. So like, for example, in guide report, we are making this simple like metaplot of activity of that guide across all kinds of loci and all kinds of experiments. So this is for it, so that we can align everything, and we are just showing the events. Let me show you on some report. We had here reports, and we can go open the one by guide. Oh, maybe not. Maybe we'll open the real one from here. Guide report. We are running low on time, I see. I was not prepared. Yeah, so this is basically summing up all kinds of deletions that this guide does across all kinds of sites and conditions. And you can see it on one figure. So, sometimes it doesn't look very pretty. So these are deletions. And this is my variant plot, which shows you what kind of reads you have there. But this is summarizing all kinds of experiments here. And then you have also this editing rates and so on. So this is the guide report. And yes, if we continue, this is about normalization now. Yes, so this is very important, how the normalization works, because we are working on the event level. That allows us to be more precise at normalizing. And here's an example. Normally, how they normalize, like I think how they normalize in CRISPR, for example, is that you've got this thing, you see, and you see that in the control, and then they will just subject this reads, okay, we remove this menu, right? So it kind of decreases this. What we do is we have a threshold, and if events in control are above this threshold, then this is assumed to be of the background, genomic background. And then that means these events, these deletions, should be completely removed from this treated sample. And this one is also genomic background, so it should be completely removed. And this is what happens in ampliCan. It will be completely removed, and only the ones that don't have anything in the controls are left. This is how ampliCan normalizing works. And also, you have this option here. By default, we are normalizing. You specify here control column. You specify 1, for example, and this is control line. And then you additionally need to specify this normalize. By default, it is using 2 columns, guide, arena, and group. So if we print pipeline, so by default, this has somewhere over here, you will see that this by default has this guide, arena, and group. So like normalize. You would have it like this, okay? I don't know. And group. So that means if you grab specify this control as 1, then we will look inside the columns guide RNA and group together. So this guide, and for Betty only, which other experiments don't have control specified for Betty and guide RNA. And we will use this to normalize every other row which has the same Betty and this guide. And if we specify it for Tom, then it will look for this guide and for Tom. If we make it simpler, for example, we want to normalize not by guide, but by maybe just group, then you just remove it like this. And then you specify Tom is going to be 1. So then we are looking, okay, this is control for a Tom group, where in the Tom there is editing. This one is going to be normalized by this one. Or you can specify just by guide. Guide, I don't know. This is rather confusing for people, but this is a powerful way to kind of match which samples can normalize which samples. So you can specify also by ID. If you specify here by ID, that practically disables normalization, which is the same as when you null. And you don't normalize. So I don't think you can normally check your events in the controls using this special variant plot. So you can run without any normalization. And for example, here in the variant plot, oh no, not this one. Not this one. Here in the ID plot. So you go into report breakdown by the ID. And here you can see something like this. And this is clearly a control. And if you see nothing suspicious in the controls, then you know, okay, do I need to normalize or not? So in this case, we have something suspicious here, which is not visible on the plot. Oh, no, it is here. You see? Let me zoom in. Here is this variant plot. So this shows you the most frequent reads across the region of interest. And this is the amplicon sequence, and you can see the frequency at which we find it. And you can see here that this guy, this person in the control, has 50% of the reads have this A instead of G. And this is an event that we could normalize for if we want to. But sometimes we don't want to do that because this might be normal for mosaicism of a patient. And maybe we want to keep it. So it's up to you. So I think, yes, then we have, if we are going to different kinds of plots, so I can explain also, this is the deletion plot. So this arc here is a deletion. The highest is normally in CRISPR, you can see here the third base. So this is the palm. The third one from the palm has usually the highest. You see, the single base per deletion usually has the highest frequency. And then every guide has its own sort of pattern that they like to make. And then there are some also large, very infrequent ones. Maybe primer dimers, not completely fitted. These are insertions. So insertions over here you can see. And mismatches. And it matters at which frequency they occur. So you can see in our case, this is the patient who has 50% almost of this G to A transformation. So yeah, because this is some real data. I think we are need to kind of rush it now. So we are still at the end. So then it's a summarization of everything, of all the events. This is called the software works. It summarizes the reads. And yeah, and you get the table and you need to calculate sort of your own edit rate and HBR percentage. Normally, we divide by reads filtered columns. So reads edited divided by reads filtered, or HBR reads divided by reads filtered to get these percentages. And then at the end, this sort of produces you all kinds of reports. I have shown you some of them and links to all of the other reports. Then we've got the barcode report, which shows you the multiplexing, how it went. Then you've got these meta plots. So these are the least sort of, these are just like interesting to look at, but usually not interesting for your experiment. Unless you specify in some interesting groups, maybe you can see which person has the best skills in the lab or something like that. Or maybe which amplicon sequence works best for you, or which guide works best for your amplicon, things like that. And then you've got the ID report, which is the main one, right? So the ID report is the thing that we should always reference to, and then take a look at each specific site. And then I wanted to say one more thing, because these ID reports are done let's open one of those. Here you can see they are basically RMD. So it's just possible that you want to adjust things, so you open that. You open that, and you have params. This is which paths are loaded for the files at the top. This is the params, and this is, you can see, you can replace this stuff here, and then start tweaking, and you can look internals how everything is being plotted. So this shows you how to make the distribution plot. We just grab the config, we factor on this, and so on. And this is a plot. And then how to calculate the percentage of frame shift. This is how it is done. This is edit rates. Maybe you want to change this. Maybe you want to round the numbers, so you can add here round on top of it. And now you're gonna have nicer numbers for the report. And you just click neat, and that's it. And it will regenerate the plot for you, the report. And then you can open it as HTML. So this is the main sort of story, and I haven't even started with how to use these LLMs to make the configs for you. So we can close this. I think we can close this. But I think you can look it up yourself if you are interested in doing that. Because now I have published these vignettes. So one is about tweaking the outputs. And here you've got like a strategy how to do it. Normally, I recommend you to sample, because ampliCan is slow, but there is an option to sample. And you can sample like 10,000 reads, produce quickly results. They are not going to be perfect, but they're going to give you an insight into how well it demultiplexes the files, how well it assigned each ID, and then how well HDR is being quantified, and
You're breaking again, Kornel. Nope, we cannot hear you.
You cannot hear me. Give me a second. I'll try to open here. That will help. Is it better now? Can you hear me?
Yes, we can hear you now.
Sorry for the quality of the internet. I didn't have at the office today. Okay, but yes, I was saying that I usually recommend to have a separate folder for every output for all kind of parameter group, and then you can compare the results. So take a look at this vignette if you want to tweak the outputs. Then there is a vignette for creating a config with the LLM. And the main thing here is that you don't want the large language model to directly transform Excel into a config. You want the large language model to write a script that will load the Excel file, transform the columns using code, and then produce the config. In that way, you can check the script yourself and confirm that it works correctly. And there are some strategies how to do it here, described. So this is probably the least painful way to do it, because making config can be really, really difficult and painful, depending on what you get from the biologist, from the lab. So I recommend to take a look. Also, there is like a prompt which you can use for those models that will help you build the config. And there are some examples of things what to do. So, for example, uppercase by guide with some buffer, or uppercase the full interior, and so on. So take a look at this. And then there is the code explained also vignette. If you are interested to see exactly how it works. And I think this may be sort of reaching the end of the seminar. So this was the ampliCan package. I guess now we can address your questions if you have some.
Thank you so much for that wonderful presentation. So now we'll be taking questions. In case you're a bit shy to unmute and ask your questions, we'll be taking them on the Frama pad from line 60, but you can add yours on line 63. But if you have questions, please feel free to unmute and ask Kornel directly. So I will read the ones that have been posted on the Frama pad thus far. I think this one was mine, and I was asking if when you're building the config, because you had said to capitalize the guide, but in another example, you did show the guide and the PAM capitalized. Do I also need to capitalize the upstream and downstream, probably like 10 bases when specifying that?
So there is this option for this option called buffer. So I depends what you like. I normally uppercase just the guide, and then I add the buffer to be 10 base pairs. So there is this option here. It's called buffer, cut buffer. You see, it is 5 by default. I make it longer to 10, and I that's normally what I do. But it's entirely up to you. You can just set the cut buffer to 0, and uppercase whichever region you think should be the region of interest, sort of, where you want to detect the deletions. Yeah. So this is the option for it, cut buffer.
Thank you. There's also another question that asks, please clarify your point on primer placement to cover expected deletion when doing bidirectional sequencing. So, and they've put
Yeah, I understand. I think what is the question. So we can go back to maybe to the first. This one? No, this one. Primer assignment. Yeah, so I think it is best if you can have like either 300 base pair sequencing. For example, if you want to cover 300 base pair, you do 300 base pair sequencing. Or your guide is ideally in the middle. You want to have like you want to assume that the guide will cut, and that the cut will mess up this region. and you need to sequence past it. So, and you need to have quite a bit of sequencing like on this side so that it can align correctly. So preferably, it's best if you can even grab the other primer, but it's not always necessary, right? So yeah, so you can also try to align to the genome and then treat your reads through that way. But mostly I try to, if for example, some people make a mistake and then they put their guide on this side, very close to the primer, and then this sequencing cannot reach well past the guide with the deletion, and then the forward primer is also might be deleted, and then this whole thing is completely difficult to do anything with it really. So like the ideal cases are guide in the middle, you are sequencing with enough depth, enough bases that you go past it and even reach the other primer. So that is my ideal sequencing, 150 base pairs that reads from here to here. Okay, I hope that answers the question.
Yeah, I hope so. Another question is, please clarify if ampliCan does actual demultiplexing of pooled samples directly from sequencing and also performs QC.
Yeah, so the QC is you have to do it yourself using some like the fast QC. You normally run some other tools for that, which do it much better for sure. And then you can see the QC a little bit on those reports, like the example one that I had here. Where was it? Like this report, the index report normally has some sort of QC, but it's not what you would expect. So Bioconductor doesn't want to load this, but it doesn't really have in-depth QC about the quality of the fast QC itself. So you do that separately using dedicated tools for that, and then you've got two kinds of multiplexing. So first multiplexing is using I7 and I5 indexes from which are used kind of commonly, and then this is normally demultiplexed by the core facility. So they have those I7 and I5 indexes, and then they demultiplex based on that. But we can have additional demultiplexing using primers inside ampliCan. So this is the part which ampliCan handles. So there are two kinds of demultiplexing, depends which kind you are using, and depends on how you are sequencing.
Would it also demultiplex using barcodes, or that you'll have to use a different tool, for example?
No. Normally, this is done by sequencing facility. So the only way ampliCan is demultiplexing is using the primers. Okay.
Yes.
And I7 and I5 is handled normally by the sequencing facility when they do the BCL2 fast tune. This is like the standard way they do it.
Yes.
Yeah.
Okay. There's also another question on the from part that's not completed, but the question is, what would you recommend for polyploids in terms of the normalization for control?
Yeah, this is tough. I recommend to, if you're running ampliCan, don't do set controls to nothing. So like 0, control column 0. And then in the variant plot, you can see what is going on. And then you need to write a custom script. And you can load events the same way I'm loading them in these reports. So you can take a look at how it is done, or you can ask your large language model to do it for you. It will show you how to load the event files. It is, for example, in this report So it is in the ID report. It also loads events, and you can see how to do operations on them. This is just a table, and then you can start making very customized normalization for yourself. I think this is the only way to deal with with these such problems.
And I do have one last question because we're already past 5:30. But if you have two separate guides that are really far from one another, so you have like a 300 base pair amplicon, you've done around 400 base pairs, sorry. Then you have your short reads, which are around 150 base pairs. When you're creating this config file, and you have two or more guides for the same amplicon. So how do you specify that? Do you create each line, each event with a separate
So
guide
here,
and separate group?
these are two separate guides, yes? But is this a new case or not?
Come again?
Is it a new case experiment or not? Are they supposed to cut like this and create a overhang type of cut, like single strand on each side? Or these are two separate guides that are just cutting?
Yes, there are two separate guides that are just cutting.
And they are very close to each other. And these are
No,
completely
they're
separate
not close.
experiments, or they're cutting the same sample?
They're cutting the same sample, but they're not close to each other.
I see. Yeah, this is difficult case. I would recommend to use nanopore sequencing for that. And then if you use nanopore sequencing, you can have long primers. You specify very long primers, and you do this nanopore enrichment. You do PCR on those long primers, and you sequence that. And then in ampliCan, you would have just single row, which would be very, very long. And you would work on like a very long amplicon, which would be, and you can one the other guide. So like all of this region that you are thinking is going to be cut, make it uppercase. And in that way, you would be able to see exactly what is going on.
All right. All right. Thank you so much, Kornel, for that wonderful presentation. In case anyone has any further questions, just add them on the Frama pad and we'll send them over for answers much later. So this session has been recorded and it will be uploaded on the Bioconductor YouTube channel today. Actually, right after this session, it will be uploaded. Yeah, so if you've not joined our mailing list, that is on line 69 of the Framapad, please feel free to sign up. And if you've not joined our community channel on Zolib, please also check line 67 of the Framapad and you will be able to see that. And now I'll pass on to Maria for final comments before we close.
Well, I just want to say thanks so much, Kornel, for a great session, and thanks everyone here for joining. And we look forward to seeing you at the next one, which is GWAS in July. So thanks everyone. Have a great rest of your day.
Thank you. Bye.